Hello everybody, I hope you're all doing well and staying safe. So these are going to be the final two, or at least my final two lectures for 703. After this you're going to have Aviv Regev for the rest of the semester. And so for the last two lectures we're going to talk about transduction and then conjugation. So for this lecture we're going to talk about transduction. Last time we talked about transposition, so a transposon moving or disrupting genes and the use of a transposon as a potential mutagen. Here we're going to talk about viral transduction. So transduction simply means viral infection. Today we're going to talk about phage transduction, but transduction will refer to any viral infection. It could be a coronavirus infection, it could be a retroviral infection. In this case, we're going to again talk about phage, specifically P1 phage. Last time we talked about lambda phage. And we're going to use transduction to do two things. One of them is to move mutations. So move them from one bacterial cell to another bacterial cell. And the second is essentially to map mutations. So and here we're going to use TN5 as a method for mapping mutations and even sort of trapping them in place. Okay, so the way that we're going to use viruses here is to actually use a viral packaging mechanism or machinery to actually move genetic material from one cell to another. So in the last lecture, we talked about infection of bacteria cells with phage. In a bacterial infection, you have a phage particle that's associated with a bacterial cell, and it inserts its genetic material into the target cell. This DNA is then copied. In the case of P1 phage, P1 phage is about 100 kilobase phage particle. You have constant replication of this phage DNA by what's called rolling circle replication. So basically, the phage DNA circularizes and continues to replicate again and again and again and makes tons of copies of its DNA. This DNA is then encoded into the phage packaging or the phage capsid. So you have the introduction of phage DNA back into these capsids, and then you have release of phage out of the cell during lysis. So you have tons of phage particles that are released following the lysis of the cell. So what's interesting in the context of phage packaging is every once in a while, the phage essentially makes a mistake and actually packages not its own DNA, but the bacterial chromosomal DNA. So the other piece of DNA sitting in the cell besides this rolling circle replication of phage DNA is the E. coli chromosome, or the bacterial chromosome. This chromosome is around 4.6 times 10 to the 6 base pairs, so around 5 million base pairs. It also is replicating, and every once in a while, a piece of this bacterial chromosome is actually packaged into a phage. So how often does this occur? Well, it occurs about 1 in 300 phage particles contain bacterial DNA. So 1 in 300 times, instead of phage DNA, these phage particles contain just a piece of bacterial DNA, no phage DNA, just about a 100 kilobase chunk of phage DNA. So you can imagine if we divided this bacterial chromosome into around 100 kilobase pieces, we'd have around 50 pieces of chromosomal DNA. And so say we had a TN5 insertion somewhere in this bacterial chromosome. Here we have TN5. Remember, TN5 generally has a resistance gene like kanamycin resistance. So we have this 5 kilobase TN5 plus kanamycin resistance. And in 1 in 50 events, the bacterial chromosome or piece of DNA that's actually packaged will contain this TN5 insertion. So 1 in 50 will contain TN5, just like it would contain any bacterial gene of interest. And so roughly 1 in 300 times 1 in 50, so 1 in 15,000 phage will contain TN5. So we can actually use this system, this sort of mistaken packaging of chromosomal DNA, to actually move TN5 from one cell to another. So why would you want to do this? Why would you want to move TN5 from one cell to another? Well, say you wanted to actually introduce a mutation onto a different background. You could do that simply by moving cells using a phage packaging system, from moving DNA from one cell to another. Okay, so how do we actually do this experiment? Well, say we actually have a bacterial chromosome that has a TN5 insertion into a particular gene of interest. So say this is gene A, and it's disrupted by this TN5 insertion. And we want to move this from one bacterial cell to another. Well, this TN5 insertion is on a bacterial chromosome, and so we can simply take this bacteria and infect TN5 mutant cell with P1 phage. Then we can collect virus particles. So we have the phage going in, packaging its own DNA, and then every once in a while it's going to package a fragment of DNA that contains TN5 and this disruption of the gene of interest. And then we will use this virus to infect another set of bacteria. So following infection of this other bacteria, we're going to select for an event in which TN5 has inserted into the DNA in the opposite cell, or in the infected cell. So what does that look like? Okay, so here we have a piece of DNA that has TN5, and this piece of DNA is coming from a phage particle. So it's been accidentally packaged into a phage particle. So this is essentially P1 derived, and we've infected it into a target cell, and the target cell has a bacterial chromosome. And once this piece of DNA is in that target cell, it can undergo recombination with this bacterial chromosome. So just to remind you, all of the sequences outside of TN5 here are essentially bacterial sequences. So they share identity with the target bacterial chromosome, And so you can have recombination between any piece on this DNA and the target DNA. The only region that isn't shared is the region that is just TN5 itself. So you can have a recombination event that introduces this piece of DNA into the target cell. And you can actually select for the presence of TN5 integrated into the target cell by selection with kanamycin. So TN5 carries the kanamycin resistance gene, and we can select for this event by simply selecting with kanamycin. So what happens if we don't have recombination? Well, what happens is that this piece of DNA that came in on a virus particle essentially gets lost. It can't replicate itself. It's just a piece of DNA with no origins of replication, no ability to maintain itself, and so it's lost if it doesn't get integrated into the target genome. So why doesn't TN5 jump? Why don't we use the same transposition mechanism that we talked about last time and rely on TN5's ability to move as a mobile genetic element? Well, it could, but remember, TN5 jumping occurs in 1 in 10 to the 5th cells. So transposition is a very rare event. Recombination occurs in 1 in 10 cells. So this kind of recombination that can bring this whole piece of DNA into a target genome is actually a very common event. So here we can simply select for kanamycin, or with kanamycin, select for kanamycin resistance, and know that we actually now have a bacterial chromosome that has our disrupted gene with TN5. So we've been able to move a piece of DNA from one target cell to another. So in addition to moving a mutation from one background to another, where TN5 has inserted into a particular gene, we can actually use TN5 as a vehicle to bring mutations that are adjacent to it. So where TN5 hasn't inserted into a target gene and mutated it that way, but rather TN5 actually sits adjacent to a particular mutation of interest. So let's just say we have a bacterial chromosome and we have TN5, and TN5 is located next to a mutation in a gene that's important for bacterial cell motility. So we'll call this mutation MOT-A-. So typical bacteria, when it forms a colony, will start forming these little satellite colonies outside. And so this would be a, this will be a MOT plus bacterial colony. But some bacteria lose this ability, and the cells don't migrate any longer, and so this is MOT minus, and so this MOT-A- strain is one of these MOT minus strains. So it's something we can screen for. We can actually look at bacteria and see have they gained the ability or lost the ability to move or migrate. So say we have a bacteria that has TN5 next to this mutation. And I'll talk at the end about how we actually generate this kind of proximity between TN5 and a particular mutation of interest. So we can actually move this piece of DNA from one cell to another using the same strategy that I just talked about. So we're going to use P1 to actually transport this chunk of DNA into a target cell of interest. So how do we do that? We infect our TN5 MOT-A- cells with P1. We collect virus, so after the cell lyses, and we're going to try to get this rare event where the virus has actually picked up or accidentally packaged this small piece of DNA. We're going to then infect a MOT-A plus bacteria. We're then going to select for kanamycin resistance. So select for can resistance, and then we're going to screen for cells that have lost the ability to move. So we're going to screen for this MOT minus phenotype. Remember, screening means looking. Selection means we're putting them in a selective condition where only cells that have acquired some alteration or a resistance gene can grow out. So selection, we can select tons and tons of bacteria for very rare events. Screening, we're looking at hundreds of different bacteria to look for a particular event of interest. Okay, so what does this look like in the target cell? So if we have TN5 and we have MOT-A minus, in the target cell we have a bacterial chromosome, and this bacterial chromosome has a wild type version of the MOT-A gene. And so when this piece of DNA comes into the target cell, at some frequency, you'll have a recombination event that essentially swaps this entire chunk of TN5 MOT-A minus into the target chromosome. So you have a recombination event that actually replaces this MOT-A plus with a MOT-A minus. And so the resulting bacterial chromosome will have TN5 and be MOT-A minus. So you can have this recombination event. Of course, you can have other recombination events. You can have a recombination event that occurs here, or maybe one that occurs here. In both of these cases, you're actually bringing over TN5, so you're conferring can resistance on the target cell, but you're not bringing MOT-A minus. And so the frequency with which you actually bring these two together, you bring TN5 and MOT-A minus into the target cell, or you recombine them in, is a function of the distance between TN5 and MOT-A. So if they're really far apart, then generally you'll have recombination events between these two and just TN5 going in. Whereas if they're really close together, then generally you'll actually have TN5 and MOT-A minus brought together into the target cell. So just like recombination distances we were talking about before, with linkage analysis, if they're far apart, you generally have recombination between. If they're really close together, you tend not to have recombination between. And so we can think about this as a percent co-transduction, meaning they are both brought together. What percent of time are TN5 and MOT-A minus move together into a target cell as a function of distance, where the percent co-transduction is inversely correlated with distance. So the closer they are together, the more co-transduction you have. The further they are apart, the less co-transduction you have. And so this is really a metric of genetic distance. So percent co-transduction is a genetic distance. Since we're simply asking what percent of time is there recombination between two different loci. And so what if we have a new mutation, a new MOT mutation And we want to know, is this new MOT mutation MOT A minus? So say we have a new mutation and we're going to call it MOT X minus. And we want to know, is MOT X minus equal to MOT A minus? Are they mutations in the same gene? So the way we can do this is we can actually bring over MOT A into a MOT X deficient cell and see whether we ever get recombination events that restore motility. So let's start with a MOT A plus strain where we have TN5 and we have MOT A plus together. So we're starting with this bacteria, and we will infect this strain with P1 so that we pick up lots of pieces of DNA, including a piece that contains TN5 and MOT A plus. We collect virus, and then we're going to infect our MOT X minus strain. Following this infection, we will select for the recombination event. So we'll select for kanamycin resistance, and we will screen for MOT plus cells, so cells that have restored motility. So what might this look like? Okay, so we have our piece of DNA that's P1 derived, so P1 derived, where we have TN5 and MOT A plus. And we have a target chromosome where we have the MOT X mutation. Let's just say, for example, that the MOT X mutation is not MOT A, right? So it's a different gene, and the different gene is somewhere over here. So MOT X is there. In this context, we can get TN5 MOT A recombination into the target cell, but we're never going to get a restoration of motility because MOT X is far away from this locus. So this recombination event is simply going to transfer MOT A plus with MOT A plus and not restore motility because it's not recombining out this gene of interest. So this provides a strategy to determine whether a new mutation is a mutation in a gene that we've already established or we've already mapped next to MOT A. Now, if MOT X is MOT A, then we have essentially a situation we have MOT A minus. We can have recombination that then brings in MOT A plus into this MOT A minus locus and restores function. So it's a way of looking at gene identity. So we can do a lot of the same kinds of tricks using this analysis that we can looking at mapping as we did in Drosophila and in yeast. And so one example of this is performing 3-factor crosses. So say we have 2 different motility genes. One of them is MOT1, and another one is MOT2. but we don't know what the order is of these. So it could be Tn5 mod 1, mod 2, or it could be Tn5 mod 2, mod 1. So how do we determine the order here? Well, the way we do this is by performing essentially these reciprocal crosses to look, again, much as we looked at with three-factor crosses in Drosophila, at what is the most frequent outcome, and is the most frequent outcome consistent with a particular order of genes. So what do I do, or what do I mean by that? So we're going to do essentially two crosses, right? And the first cross is we're going to start with a Tn5 mod 1 minus strain, and we're going to collect, we're going to infect with P1, collect virus, and infect a mod 2 minus background. So we're going to have essentially this P1 Tn5 mod 1 minus piece of DNA, and we're going to have phage particles, and we're going to infect a mod 2 minus background. The second cross is going to be, we're going to start with the opposite, which is P1 Tn5 mod 2 minus, and we're going to infect with mod 1. In both cases, we're going to do this infection, and we're going to screen for bacteria that have restored motility. So what does this look like? Well, let's map out these two crosses. So one will be cross number 1, and the other will be cross number 2. And let's envision two different orders, right? So we have order 1, and order 1 will be Tn5 mod 1 mod 2, and then order 2 will be Tn5 mod 2 and then mod 1. All right, so what does this look like? Well, in order 1 we have Tn5 and mod 1 minus, and we're using it to infect a strain that is mod 2 minus. So this piece of DNA is derived from P1, and we're infecting essentially target bacteria that are mod 2 deficient. So our second cross would be, we start with a strain that is Tn5 mod 2 minus, and we're transducing a strain that is mod 1 minus. In both of these, the order is the same. It's Tn5 mod 1 mod 2, Tn5 mod 1 mod 2. In fact, we can write in the status, de facto status of the missing genes here. Here we have an intact chromosome, and so this one is mod 2 minus, so it's mod 1 plus. This one is mod 1 minus, so it's mod 2 plus. And similarly, the pieces of DNA that we're bringing in, because they're derived from another bacteria, are the same. So this one is mod 2 plus, and this one is mod 1 plus. All right, so that's order 1. Now let's think about the second order. In the second order, we have P1 derived Tn5, and instead of mot 1, mot 2, it's going to be mot 2, then mot 1, so mot 2 plus, mot 1 minus. Remember, in this cross, we're bringing mot 1 minus into a mot 2 plus strain. And the bacterial chromosome has mot 2 minus and mot 1 plus. So that's the first cross. The second cross, with the same order, we're bringing mot 2 minus in. So we have Tn5 mot 2 minus mot 1 plus. And we're bringing it into a bacteria that is mot 2 plus, mot 1 minus. So here we're going to think about recombination events and how basically easy or difficult they are to actually yield a mot plus outcome. All right, so look at this strain here. So in these cases, we're selecting for kanamycin resistance. So we have to have some event that actually brings kanamycin resistance into this chromosome. So in this case, we have an event that brings in Tn5, but we don't want to bring in mot 1 minus. We need to retain mot 1 plus here. So we need to have a separate recombination that brings in mot 2 plus. So this is actually a pretty hard thing to do because we actually have two separate chunks of DNA that need to recombine into the target genome. Whereas if this is the order, in the context of cross number 2, we need an event that brings in Tn5, but that same event can actually bring in mot 1 plus. So the target is already mot 2 plus, so the result of this recombination event is bringing combined Tn5 mot 1 plus into the target chromosome. So a single chunk of DNA coming in can make this target cell now capable of moving, so mot plus. So let's think about order number 2. In order number 2, we can bring in Tn5, and we can also bring in mot 2 plus in the same event. So we're bringing in essentially this chunk of DNA into a target cell, or into the target chromosome, where you now have mot 2 plus and mot 1 plus together. So this is pretty easy, a single event bringing it over, where in the context of cross number 2, you can bring in Tn5, but here you don't want to bring in mot 2 minus, because the target is mot 2 plus. So we need a separate event that brings in mot 1 plus. So here, this is difficult because we need two chunks to move over. So essentially what we're going to ask is, what cross yields the more mot plus bacteria? So if cross number 1 shows more mot plus than number 2, so if cross number 1 is yielding more mot plus than cross number 2, then the order is likely order number 2, because in cross 1, you just require a single event bringing over this chunk of DNA. Conversely, if cross 2 shows more mot plus than number 1, then it's very likely that order number 1 is the order of Tn5 versus mot 1 and mot 2. Because again, this requires simply a single movement of DNA, as opposed to 2. So it's very likely that order 1 and order 2 is correct if, again, cross 2 shows more positive than number 1. So essentially, this is very similar to what we talked about in the context of the Drosophila 3-factor cross. We actually hypothesize the 2 different crosses. We look at the possible outcomes and compare that with the data itself. And what we find is that the more frequent event is generally the single recombination event, where the rare event is likely the double recombination event. So again, I would remind, or go back and remind yourselves of what we saw in the Drosophila 3-factor crosses, because again, it's very similar material. All right, so before I talked about finding a TN5 that just happened to be near a mutation of interest. So TN5 mod A minus. But how do we actually find this? How do we just happen to find a TN5 that's located next to a particular gene of interest? Well, the way we do that is we actually start with the whole assortment of TN5 insertions that we generated in talking about transposition. And so as you remember, we actually infected a set of bacterial cells with virus that contained a transposon, and in this case, we made the transposon jump. And it jumped in each cell into only one space, randomly in the chromosome, but we generated a lot of different insertions. So if we looked at a lot of different bacteria, we would have lots of different insertions. In fact, we had 2 times 10 to the 4th insertions, which is 1 per cell. And these insertions were spread out all around the bacterial chromosome. And so they inserted into a number of genes and disrupted a number of genes, but there are a lot of insertions that are not in genes, but are immediately adjacent to genes. And so every once in a while, say the mod A gene is there, you have an insertion next to mod A. So we're going to actually use this whole distribution of different insertions to try to identify and actually map and essentially trap this mod A plus gene adjacent to a TN5 insertion. And so how do we do this? Well, we start, as you might imagine, by infecting the TN5 insertion library. So all 2 times 10 to the 4th different bacteria, we're going to use them all and infect them with P1. We don't have to do this separately. We don't have to infect each bacteria with P1. We can actually put them all in a flask and infect them all with P1. So they all get infected, and they all start producing phage. Most of the phage contains actual phage DNA, but remember, 1 in 300 contain chromosome DNA from the bacteria, and 1 in 1,500 will actually contain this stretch of interest, and perhaps this stretch of interest immediately adjacent to a given TN5 insert. So what are we going to do? We're going to take this and take our phage that are lysing out of the bacterial cell and infect a mod A minus bacteria. So what's going to happen? Well, here we have TN5, and every once in a while we have TN5, a piece that is TN5 that's sticking next to a mod A plus gene. And in that case, this is the P1 derived fragment. When it recombines with the target bacterial chromosome, you'll have recombination that sticks in this entire piece. Now remember, in most cases, the TN5 fragment doesn't contain mod A+, but we're just looking for that rare variant that does. And so here we're going to select for kanamycin resistance. So select for an event in which TN5 is brought into the target chromosome, and we're going to then screen for MOT+. We're going to screen for a recombination event that brings in a wild type version of this MOT A+ gene into this target cell and replaces MOT A- with MOT A+. So restores the ability to move. All right, so what do we have then? Well, what we have then is we have a bacterial strain that has TN5 and MOT A+. Well, so how do we get TN5 MOT A- like we wanted to begin with? Well, as you might expect, it involves another phage transduction experiment. So we're going to take this strain and infect it with, we're going to take the resulting phage and we're going to infect again a MOT A- strain. Right? So here we have TN5 MOT A+, and we have a bacterial chromosome that has MOT A-. So what is the event that we're looking for? Well, we're looking for an event here that brings in TN5, but doesn't bring in MOT A+. So we're going to have a recombination event. We know we have TN5 now next to MOT A+. We have MOT A+ trapped, so we have it stuck next to TN5. And so we know that by introducing it into a MOT A- strain, we're going to get some recombination events that bring in just TN5 into the target chromosome. And these are going to result in a bacterial strain that is TN5 MOT A-. So that's what we want. Now we have our mutation trapped next to TN5. We can use this to move into a variety of backgrounds. We can use it to actually identify a mutation of interest. Right? So say we have some mutant in a target cell, we want to know what it is, we can actually map it essentially by localizing it next to a TN5 insert. So we know that we've introduced this proximal to a mutation of interest, and so the mutation has to be near TN5. The frequency with which this occurs, of course, is dependent upon the distance between TN5 and MOT A, and so by actually determining the number of recombinants that we have, by determining the co-transduction efficiency, we can actually calculate the distance between TN5 and the mutation, which gives us even greater resolution for figuring out where that mutation is, and again gives us an ability to now perform a lot of genetic tricks using this kind of movement. Now I'd remind you that, why do we do this, when we have other tools available? Well, for a lot of bacteria, this kind of movement of DNA is really critical to being able to examine kinds of bacteria that are what we call not tractable So where we lack a lot of genetic tools to do other kinds of analysis. So this is still very highly relevant. And I would also remind you that this is actually the way that movement of DNA occurs very frequently in this very careful interplay between phage and bacteria. And so next time, we're going to take a next step and talk about another mechanism of genome communication between bacteria, and that is conjugation. So see you next time.